In the last class, we were discussing here the early development and introduction to the central nervous system and the peripheral nervous system. Now, we have already told you that the entire nervous system in the human being or in any vertebrate, it develops from these three elements. This is called the neural tube, and these are the two columns of neural crest cells standing on either side of the neural tube. And you have already seen here that the upper half of the neural tube will form three brain vesicles, and they will actually develop into the forebrain, midbrain and hindbrain respectively, and the rest of the neural tube will develop into the spinal cord. And two neural crest cells columns are standing on two sides of the neural tube. And now these three elements are those which in fact will give rise to the entire nervous system of the body. Central, peripheral, whatever functions will develop, there will be no fourth thing at all. Only these three things are there. Bilaterally you see that they are controlling the two sides of the body. But you see here this neural tube is not just, it's a neural tube. That means it has got a canal in the center, and that canal is supported by the walls. And walls throughout the entire thickness, through the entire thickness means the entire thickness of the wall is made up of the epithelial cells. We call them the Neuro Epithelium. This is the entire thickness that actually occupies the entire thickness of the wall of the neural tube, right from the cephalic till caudal region, full thickness, in fact it will comprise of the neuroepithelial cells, and in the center is lying the central canal, and the central canal will be present here in the spinal cord as well as in all three brain vesicles. And in the brain vesicles they are more expanded, that is why they are called the ventricles, and the spinal cord is more canalicular, you call them the central canal. And on the two sides you know that you have got the neural crest cells. We do not have any fourth thing at all, nothing is hidden, everything will develop from these three elements. Now, if for example you are watching in these three elements, that means we have got actually the central canal, we have got the walls composed of the neuroepithelial cells. We are now having a cross section or the transverse section of these three components here in the middle now. And in these three components in the middle means in the middle will be the part of the neural tube. This is the neural tube which has been actually drawn here. You see this blue color, all is the neural tube, but it's the neural tube, and in the center of the neural tube this is now the central canal. And central canal is surrounded here by the walls. This is the wall of the neural tube, which is through the entire thickness, it is made up of the cells. These are called the neuroepithelial cells. You see this neuroepithelial, and they occupy the entire thickness of the wall. This is neuroepithelial cells. This whole is the neuroepithelial cells, and this is now the central canals. Now, this area which I am actually telling you, this section has been actually cut in the middle of the neural tube. Means in this section means this should be the spinal cord. And similarly, this thing actually will go to the cephalic region. This will form the brainstem, and still more rostrally or cephalically it will become the cerebrum, I say it will become the forebrain, etc. Now, see this area. Now, these are the neuroepithelial cells. These neuroepithelial cells now will form, initially they will form the primitive neurons, or you can say neuroblast. They will differentiate into the neuroblast, and they will differentiate into the neuroblast, and proliferation is so fast that they in fact will be dividing at a very faster rate, and they will accumulate themselves here around the central canal. You see, these are the neuroepithelial cells due to huge proliferation, enormous pace at which these cells are differentiating into the primitive nerve cells. You call them neuroblast, and these neuroblasts will form the neurons. And once they have been actually been forming the neurons, you will see that they will forming the neurons, they will gather themselves around the central canal. and once they have actually been converted into the neurons, their dividing power will cease. They will not having any mitotic divisions. So they will get here, you see that they are surrounding it. So they are surrounding it, surrounding around. So they will be actually gathering themselves around the central canal. When they gather themselves around the central canal, you will see here two zones will actually be developing as a result of this. Two zones will develop now. Now see, these are two zones which are actually developed. These are all neuroblasts, they are the neurons now. Now this layer which I am actually telling you here, I said this layer which I am telling you, this is in fact arranging itself around the central canal of the neural tube. This layer is called the mental layer. And the one which is devoid of the neuroblast or the nerve cells here, this is called the marginal layer. So we have the two zones now, the marginal which is in the margin, the mental which is in the center. But in the center of this mental layer, we have the central canal. And these neuroepithelial cells have actually given rise to these neuroblasts. And now they are the mature neurons. And these mature neurons, they lie in the mental layer. This mental layer will be called as the future's gray matter, because it is containing here the cell bodies here. But you see that it's just initially these are the neurons which are in fact having no cytoplasmic processes here, no dendrites, no axons. So they are, you see, they are apolar, and then they will actually give the cytoplasmic extensions here. Some are called dendrites and one will be called as the axon. These are the neurons which in fact will develop here. We see that. But you have got the marginal layer and you have got the mental layer. The mental layer is the future's gray matter and marginal layer will be future's white matter, because it does not actually have any cell body present here. These are the areas where the cytoplasmic extensions, you see, dendrites or axons, they will be actually running inside them. And because the axons and dendrites are the fibers, they are called the neurites. If the neurites in fact are surrounded by the myelin sheath, that is why they look actually in sections they will look white. They are running in this zone now, in a marginal zone. That is why the marginal zone will be called as the white matter. So we have the gray matter, central gray matter and peripheral white matter. In all the parts of the central nervous system, centrally will be the gray matter that will be surrounding the central canal, and peripherally will be the white matter, until there will be the two components of the central nervous system where the prefix will be seri. When the seri word comes, you see the seri prefix will come, that is called the cerebrum or cerebellum. Here the gray matter will be peripheral, gray matter is outer and the white matter will be inside. Inner will be the white matter and outer will be the gray matter, only in the two components of your central nervous system. One is cerebrum, another cerebellum. That means the gray matter of the cerebrum is peripherally arranged. Because it is peripherally arranged, white matter is centrally arranged. So white matter will be surrounding the central canal. In case of only cerebrum and cerebellum, though the cerebellum does not have any central canal. So centrally available white matter and peripherally arranged gray matter, and that peripherally arranged gray matter only in the two components, I say only in the two parts of your central nervous system. One is cerebrum, another is cerebellum. Because this gray matter is arranged in the periphery, that is why it is called the cortex. You call them cerebral cortex, because the white matter is arranged in the center. That is why the white matter can be called as the medulla, but usually we do not use the word medulla too often. You see, this is called, so white matter of the cerebrum or white matter of the cerebellum, both can be called as the medulla of the cerebellum and medulla of the cerebrum. but we usually avoid using the word medulla for the central white matter of the cerebrum and the cerebellum. But you see, this is here the mental layer, we have the marginal layer, we have central canal. After your mental layer has given huge amount of 100 billion neurons are actually present here in the mental layer of your central nervous system or the gray matter of central nervous system. 100 billion neurons. If this is actually here, so and they are derived from the neuroepithelial cells. Once the neuroepithelial cells have ceased to give now any more neuroblast, it will now give rise to neuroglial cells. These are supporting cells, you see, kind of a connective tissue of the nervous system. Supporting cells. The two important types of neuroglial cells will be created by the neuroepithelial cells after they have completed their pool of the nerve cells. They have given their nerve cells, and now it has ceased another process of giving a derivation of another group of the cells will be actually given. This is called the neuroglial cells, and these neuroglial cells will be in fact present here either within the central nervous system, I say within the mental layer. Neuroglial cells will be present, and these are called astrocytes. These astrocytes will be actually the supporting cells. These are they are supporting the neurons and the blood vessels, which are actually running running among the mental layer or among these neuroblasts here. These are called the astrocytes. But at the same time, we have got another group of neuroglial cells. These are called the oligodendroglial cells, and these oligodendroglial cells will actually be present here in the marginal layer. Why they are present in the marginal layer? In the marginal layer, because in the marginal layer are running now your neurites or the fibers. So these oligodendroglial cells here, oligodendrocytes, they will form the myelin sheath for the neurites which are running here in the marginal layer. And these neurites which are running in the marginal layer will actually be provided with the myelin sheath coming from the oligodendroglial cells, and the oligodendroglial cells again come from the neuroepithelial cells. But we don't have just the oligodendroglial cells in the marginal layer. Within the mental layer we have got also glial cells, but these are called astrocytes. So astrocytes and oligodendroglial cells, these are the glial cells coming from the neuroepithelial cells after the neuroepithelial cells have completed their differentiation into the nerve cells. After they have given 100 billion neurons, now the next population of cells which will be derived from the neuroepithelial cells are the neuroglial cells. Within mental layer will be the astrocytes, and outside mental layer forming the myelin sheath for the neurites which are running either from lower region from spinal cord to the higher centers, or from higher centers to lower down into the spinal cord, running up and running down. This means it can be going up from below up or coming from above down. These are the neurites, these are the bundles of the fibers which will be actually running up and down, and they are usually myelinated. And this myelin sheath that has been given to these bundles of the fibers is coming from the oligodendroglial cells, oligodendrocytes, and the oligodendrocytes basically comes from the neuroepithelial cells. And after the neuroepithelial cells have given you 100 billion neurons and as many number of oligodendroglial cells as well as astrocytes, the third population of cells, after they have ceased to give these structures or these cells, the third population of the cells will now be settling here. You can say settle around or settle at a margin between mental layer and the central canal. So you can say they are forming here the epithelium of the central canal. This is epithelium of the central canal which is being formed by the neuroepithelial cells. Third attempt to form the cells by neuroepithelium is these cells which are lining the central canal. These cells are called ependymal cells. So you can say ependymal cells will be the third population of cells coming from neuroepithelium. And at the same time, you have seen the second population of cells is the oligodendroglial cells, astrocytes within mental, and oligodendroglial cells. Means these are the glial cells, inside will be the astrocytes, and outside here in the marginal level will be the oligodendrocytes. Astrocytes, oligodendrocytes together call them glial cells. This is the second population of cells, and you know that already in the mental layer is lying the first population is the important one, that is called the nerve cells, or you can say neurons. These are the three different population of cells. They have been actually given to you. Your entire nervous system, central nervous system, has been decorated with the three different types of cells, but all coming from neuroepithelium. At the same time, you have seen this area. This is what we call it. So entire arrangement right in the spinal cord, in the medulla oblongata, then in the pons, then in midbrain, at least in the spinal cord and in the brainstem. We told you that medulla oblongata, pons and midbrain together is a single unit. We treat it as a single unit. These are three different components of central nervous system, but we will actually be discussing these three components as a single unit, and that single unit is called the brainstem. So in the brainstem and the spinal cord, almost arrangement is same. You see that because the spinal cord and brainstem considered as like a single organ. I can even discuss the spinal cord and brainstem like together as a single unit, but usually we discuss spinal cord as a separate thing and brainstem as a separate thing. But most of the features they are identical. Here central canal, they are also central canal. We call their central canal in the brainstem as ventricles, but here we call them central canal as central canal. Mental layer here, mental layer there also in the brainstem. Marginal layer here, marginal layer in the brainstem also. Though arrangement in the diencephalon and cerebral hemispheres and in cerebellum can be little different, because you know that gray matter is here inside, I say centrally available gray matter, and peripherally white matter, but in the cerebrum and cerebellum this is the other way around. Gray matter is peripherally arranged and white matter is centrally arranged. But you see here in this area we call this as the arrangement here. Now in this area, if this is the central nervous system configuration, how the peripheral nervous system develops? You know that this is the mental layer, and in this mental layer especially, because the central nervous system, I say nervous system, has to perform two important functions. One is called motor function, another is sensory function. As far the motor and sensory functions are concerned, so the neurons in the mental layer, after the neural tube has undergone full closure here, you see at the end of the 4th week, this neuroepithelial cells which have already given the mental layer, these cells will actually be divided into the two groups now. One will be given the assignment of being sensory in function and others will be the motor in function. And here what will happen? This mental layer's ventral or anterior part will in fact have a separate bulging. So here it will have a thickening. And this thickening is giving you the group of neurons which are motor in action. Their function is motor activity. You can call them efferent neurons. But the posteriorly available, this mental layer portion will have another swelling here, another bulging, I say thickening here. This is dorsal thickening, you can say. And their function will be sensory. You can call them efferent neurons. And similarly here also. Now you can see the mental layer again divided into two parts, the dorsal thickening are the posterior thickening, that will become posterior thickening. You call them this sensory thickening, and this plate that will actually be present here right from caudal region up to the cephalic region, means you can go the similar plate, you see that like a column it will stain now entire neural tube here. This plate will be called as the alar plate. Now this is called alar plate. And this plate which is ventrally available here for the motor function, you can call it the motor plate, you can call it the anterior plate, you can call it the ventral plate, but here embryologically this is called the basal plate. So alar plate and basal plate means this is now your sensory neurons, future sensory neurons, and these are called the motor neurons. They will perform the motor function and they will perform the sensory function. They are sensory neurons within central nervous system, they are motor neurons within central nervous system. Here incidentally in the spinal cord, but a similar thing will happen in the rest of the neural tube or rest of the central nervous system also. Specifically you see that brainstem. And if you are actually watching here, so here will be between these two thickenings, because these are the two bulgings here, so central portion will not bulge, and here is a depression. This depression is called the sulcus, because sulcus is actually this in transverse section, but this will be present throughout the entire length of your neural tube. You call this sulcus as sulcus limitans, limiting sulcus. This is sulcus limitans, limiting sulcus. Limits between a kind of a border between sensory posteriorly and motor anteriorly. Motor neuron, sensory neuron, this is called sulcus limitans. And if this is sulcus limitans, then we have got the two different types of neurons we have already noted here. Now if you are watching your motor neurons, now you see this is central nervous system that has actually been developing from these things now. But at the same time, because we have got the peripheral nervous system also, so in the peripheral nervous system, you have to actually watch here how the peripheral nervous system develops and how it is connected with the spinal, with the central nervous system, because peripheral nervous system emerges from the central nervous system. And in this central nervous system and peripheral nervous system, you have to actually watch here. Now consider now your motor neurons. The motor neurons, I told you that is coming here in the basal plate. And it is performing in the basal plate, again it will perform two different functions. It is motor, this is your workforce, you can say in your society, this is the workforce. This is sensory, this has actually been placed posteriorly, it will stay now, it will stay inside the central nervous system, it will not come outside, it will remain inside, just like your in society the women has to stay inside the house. But this is the workforce, you see that, the male. They have to actually go outside for the work. This is motor. So this has to come outside, but this will stay inside. You say, sir, if it stays inside, how it will actually get the sensory information from outside? For sensory information from outside, you need another group of neurons, and that group of neurons doesn't belong to the central nervous system, that belongs to the peripheral nervous system. And if the peripheral nervous system means the sensory neurons belonging to the peripheral nervous system, they are not present inside the central nervous system, means they are actually derived from, not from the neural tube, but they actually will be derived from the another group of cells, you know, that is lying like a column outside, that is called the neural crest cells. We will come to that later, but you see here in this especially motor neurons, if this is a motor neuron, we are coming to the basal plate. In the basal plate, it will be divided again into two portions. For the voluntary, again one voluntary and involuntary. You can consider, okay, voluntary, involuntary, we divide it here in such a way, close to sulcus limitans. 1/4 you see that. This mental, I say this basal plate area, 1/4, this will be for involuntary function. But the rest of the 3/4 you can say, just roughly 3/4. So you see the rest of the 3/4 anteriorly placed here, they are for voluntary function. Consider the voluntary first. Leave the involuntary, we discuss involuntary motor activity. This is called something for the autonomic nervous system. But you watch here this system, voluntary system, somatic system you call it. But you see one neuron we are actually picking as an example. You see, as an example we are taking just one neuron. One neuron, this is one neuron here. Motor neuron, this is motor neuron. This is motor neuron, and such motor neurons, so within the central nervous system, within your central nervous system, neurons usually are multipolar. Multipolar neurons means they have got the cell body, they have got multiple dendrites from all around, and there is a central axon which is actually present here, the single axon. So on one side there is a single central process that is called the axon, and from all sides you have got the dendrites, the branched cytoplasmic extensions, you call them the dendrites here. Such neurons are called the multipolar neurons, and these multipolar neurons basically are the neurons which in fact they fill the central nervous system. Central nervous system almost always contains multipolar neurons, either sensory or motor, they are multipolar neurons. So consider here this is one motor neuron, these are dendrites, and these are now the axons. Now watch the axons. Now these axons will will enter now or leave the mental layer, will now enter into the marginal layer, will break through the marginal layer and appear on the surface of the spinal cord, surface of the central nervous system, here incidentally spinal cord. And these are the axons which are now coming out, you see. And these axons in fact will come, and these axons when they are actually coming out, and they will finally end at their effector organs, means these are the muscles, skeletal muscles here, because this is a voluntary somatic, that is why it will be skeletal muscle. But when it will be involuntary, then it will be smooth muscle or cardiac muscle or exocrine glands. But you see this is effector organ. Now this is the neuron, and this is the motor activity that is being actually carried out here. When here is the stimulus here, you see action potential here, skeletal muscle fiber will contract. This is the motor activity, and now once it comes outside the central nervous system, you will call this as the part of peripheral nervous system. And here is the axon, and once it will come outside here, you see it comes outside here. The axons here, it is not one only, it will be thousands of them. So this is now many axons are coming outside. Once they will come outside here, they will actually get the myelin sheath. Outside, inside they have also the myelin sheath, and this myelin sheath has come from the oligodendroglial cells. And these are the derivatives of the Neuro Epithelial cells. But once they have actually break through the surface of the central nervous system, outside they will again get the myelin sheath, but this time the myelin sheath will be provided by the Schwann cells. And these Schwann cells in fact will be derived from the neural crest cells, which are standing, you see, on either side of your neural tube. So this is one of the derivatives of your neural crest cells are the Schwann cells, which are giving the myelination to the axons, which have now to the motor axons obviously, which is break through the surface of break through the marginal layer and come outside here. So you will see in this area myelin sheath will be actually given to them. So this myelin sheath has been given to them. So this is the motor neuron, this is motor activity is being here. So see, this is called the motor neuron, and this is the motor activity we are watching. But we do not have just motor activity, we have the sensory activity also. So if this is now skeletal muscle, you see this is now skeletal muscle here on the surface. At the same time we have here skin, and when you are applying the stimulus to the skin, here are the specialized organs which are present here in various layers of the skin, you call them sensory receptors. And these sensory receptors which are present here, once you apply a stimulus, these sensory receptors will transduce that stimulus into impulses, and these impulses will actually be transmitted from periphery to the central nervous system, and for this purpose you need again neurites. So and these neurites are required here to carry these impulses to the central nervous system, and for the neurites to develop here, to develop or to get the neurites or to receive the function of the neurites, you need the neurons, and these neurons are the sensory neurons. and these neurons are sensory neurons, means they will actually pick the impulses, or you can say the transduced impulses which have been transduced from the applied stimuli. They have to actually carry them and will deliver them to the central nervous system, and such neurons, they are not these neurons which are actually lying inside. These are the neurons which are lying outside the central nervous system, and these neurons which are lying outside the central nervous system, because because these neurons should stand outside central nervous system, and if you need a neuron or a nerve cell outside central nervous system, the source will be only one, that is the neural cells, because neurons can develop from only two sources: neuroepithelial cells within the central nervous system, or if you need any neuron standing outside the central nervous system, then the source is neural cells. There is no third source at all. If you see any cell body outside central nervous system, means outside brain or outside spinal cord, always mark it is a derivative of this neural cells. You can say we have got 100 billion neurons. Why not some of the neurons actually come outside his spinal cord and brain to stay outside here? No. Neuroepithelial cells, we have 100 billion neurons derived from neuroepithelial cells, but not even a single neuron will leave the brain or spinal cord to get itself arranged outside them. They will remain always inside. If you need anything outside, it should come from neural cells. There is no other derivatives, there is no other derivative, there is no other source of neuron that is placed outside central nervous system except from neural cells. And here neural cells means if these receptors which have transduced the stimuli into impulses, if you want to actually pick them, so for this transmission purpose you need the neuron, and this neuron stays outside here, and outside means coming from the neural cells. So this neuron comes from the neural cells. This is the neuron which come from neural cells. And this is the neuron that comes from the neural cells, and this neuron initially will have only one process, and that one process is not enough, because if this process will actually get connected to the receptor, sensory receptor, it will actually get the stimuli from sensory receptor, sure, but how it will actually carry these impulses here to the central nervous system? It is not possible. That means you need another neurite. That is why this single neurite, that is called a single neurite here, or axon, will split into the two branches here. Split into the two branches. You see, this is a type of a neuron, different than the multipolar neuron that you have seen already. This type of a neuron which has got the single axon, but does not have dendrite, lying outside here, derived from the neural cells. You see neural cells, now column we are actually erasing, and we are just focusing on a single neuron. It is not single, there are thousands of them, but you see we take one sample of this neuron. You see this. This type of a neuron actually is present where this is for the sensory purpose. It actually receives the impulses for the first time. This is the neuron that will actually test the impulses for the first time, and this will split into the two branches. You see the one branch that will run along with the motor neurons in a same packet, that is actually, and this is, will be covered by means of covered by the connective tissue, and in the same packet it will go, and this neuron which we are actually telling you, this is going and get connected here to the sensory receptor for impulses to transmit, and these impulses will actually be carried here toward the cell body, and the second branch will in fact enter here into the posterior aspect of your central nervous system, here especially spinal cord, and here in the spinal cord it will enter and will relay its impulses in this LR or in this sensory plate here, where again multipolar neurons, they are waiting for them. Now here one sensory neuron I am telling you here. This is multipolar neuron which has got the dendrites here, and this neuron which is standing outside, which is the other branch, you see that, which has got one axon, which has got the peripheral branch connected here to the sensory receptor, and central branch which is actually coming here and entering here into the central nervous system, crosses the marginal layer, enters here into the gray matter, that is called the mental layer, LR plate or the sensory plate, where the second sensory neuron is actually waiting for them, and here it will relay into them, mostly they will relay. So this is the neuron which actually carries the impulses from the sensory receptors and transmit them to the sensory neurons actually which are present inside the sensory plate of the gray matter of the central nervous system, and it has actually entered inside here. Now you can say these are the peripheral axons. So this is called the central axon, this is called the peripheral axon, because axon is only one, we do not have dendrites, but the peripheral axons are those axons upon which the stimuli are traveling toward the cell body. That is why they are sometimes called as the dendrites also. And these are the central axons from where the impulses are traveling away from the body to the central nervous system. That is why they are called as the central axons. So axon splits into the peripheral axon, you can call them dendrites, and this will actually be forming the central axon, you can call them axons. And because they are actually here, so they will get the myelin sheath again, and these myelin sheath, you know that is coming from the neural cray cells. So the neurons are also derived from neural cray cells, and their myelin sheath is also derived from the neural cray cells. So this means the myelin sheath will actually be developed here, so myelin sheath will be there. You see, the myelin sheath is there. Now you see, this will be actually purely sensory group of neurites, and this is a purely motor group of neurites, and they both are in contact with the central nervous system. So these are called the roots also. This is called sensory root, this is called the motor root. Both have joined to form here a common packet of the neurites, you call them nerves. This is actually the nerve, and this nerve has got the sensory as well as motor fibers. And the sensory and motor fibers, they have stopped or they have finished at the neuromuscular junction. You see their effector organs, and the sensory fibers, they have stopped at the sensory receptors. So you can say your peripheral nervous system is like this, but you see this is the neuron. This neuron is lying where? This neuron is lying on sensory root. That is why this neuron, because it is not one, there are multiple discrete collections of neurons. I am actually showing you as a simple single, but you see there are many of them. So this is a discrete collection of such neurons here, and this discrete collection of such neurons on which root? On sensory root. And such collection, if it is standing outside the spinal cord, I say standing outside central nervous system, if you see a neurons collection outside the spinal cord or outside brain, such collection's name will be ganglion. And this ganglion is present on which roots? You see posterior root or sensory root. That is why these are called sensory root ganglion, because sensory root is posteriorly available, that is why you call them posterior root ganglion, or dorsally available, you call them dorsal root ganglion. These are the names which are actually given, the synonyms. And you see these are the ganglion which is actually present. So discrete collection of neurons lying at one certain place, you see discrete collection, you call them ganglion. But you see, if you are actually seeing cell body collections or the nerve cell bodies, if they are present inside the central nervous system, and obviously in the gray matter, because here one neuron I am actually showing you, but if there will be multiple neurons here, you see huge number of neurons, it is not just in this transverse section, but throughout you can say such things, such neuron collections for some common function, if they are present within central nervous system, similarly like this. But here outside you call them ganglion, inside the word will be used here, or the term will be used here, nucleus. So this is called nucleus. So the word nucleus which we have been actually using for quite a long time. You see this nucleus, something central figure within your cell here. Now this term is a different term. Nucleus means huge number of say enormous number of cell bodies, nerve cell bodies present within central nervous system for some common function. This group together you will call it the nucleus. So this is called the motor nucleus. And because this is motor nucleus is present in the anterior part of your gray matter here, you will call them, because these anterior, you say anterior horn cells sometimes also, because these are the thickenings which are called as the basal plates. They will actually extend here, will reach up to certain periphery, and they will be in form of horns. That is why these are called anterior horn cell nucleus. So nucleus and ganglion, this should be differentiated. If it is a discrete collection of neurons standing outside central nervous system, and outside central nervous system cell body always derived from neural cells, and if they are present here inside, you will say they are nucleus, and they are nucleus because they are present here, you can call them motor nucleus, and if they are present here, you will call it the sensory nucleus. And sensory nucleus, and who is carrying the sensory stimuli first? Obviously these neurons. These neurons, that is why I call the first generation of neurons, you call them first order neurons. Means sensory receptors will transmit the impulses through first order neurons, and these first order neurons will in fact hand over their impulses to the second order neuron, which are standing which is located inside the gray matter of the central nervous system, and they will be actually relaying, and if they have actually relayed into them, then this same information impulses should be actually transmitted to the higher centers for perception purpose. Perceive means you have to make you feel now. Sensory receptors were just being touched. The touch word means you are feeling the touch. This is touch, means to make you feel, means perception. And if somebody is actually applying his finger on your body, on the receptors here, this is just the application of the stimulus. This is not the touch. Touch will be when you feel it now. And the feeling in fact is the perception, and perception is done by the higher centers. And higher centers, and here if this is spinal cord for example, or this is brain stem, spinal cord via spinal nerves and brain stem via cranial nerves. If this is here, so it is not the brain stem, it is not the spinal cord that will make you feel that stimulus. That will actually send these stimuli to the higher centers, and who will send them? The second order neuron. If the second order neuron means who will? Obviously you have relayed into their dendrites, and their axons will now, and with their axons, and now these axons will actually be running where? These axons will run in the marginal layer, and they are going higher up. If they are going higher up now, from below upward here, where they will go? To the higher centers, where they will go to the forebrain. If they will go to the forebrain, so they will first relay into the diencephalon, and then after diencephalon, the diencephalon will now further project to the cerebral cortex. The sensory stimuli from the periphery will be collected here by this first order neuron, and first order neuron is standing outside here, outside the central nervous system in the ganglion. We call them sensory root ganglion, and its central axons will form here the root, again myelinated, a myelination coming from the neural cells, and will enter now into the marginal layer and will relay its impulses which it has received from the sensory receptors to the sensory neurons. These are the second order neurons which are standing, or which are located inside the central nervous system. In the LR plate, you can say, in the sensory plate, in the dorsal plate, you can say posterior plate or posterior horn of the gray matter of spinal cord, or you say posterior horn of the gray matter of spinal cord, or it will be similar thing will be present in the brainstem also, but there the nerves will be cranial nerves, and here the nerves will be spinal nerves. Here the nerves will be spinal nerves, but here actually it will be the posterior or the sensory root of the spinal nerve, and there will be the sensory root of the cranial nerves. And now they are actually here, and now you can say from the second order neurons, the fibers will now, or the axons will now enter in the marginal layer, and they will rise up here, will go to the higher centers, means to the forebrain. Passing through the brainstem will reach the forebrain, and all the sensory modalities first will report to the thalamus, except female. And it is the thalamus which in fact will project it further to the cerebral cortex, sensory area of the cerebral cortex. And here the sensory area of the cerebral cortex is the final destination which will give you a sense of touch. Here the touch, later on in some other areas will be pain and many other things also. And this is what we call here. So, you can see this is your peripheral nervous system which is present here, and peripheral nervous system you are actually watching in this way. The peripheral nervous system is connected to the central nervous system, and here you have seen these upward, you see upgoing axons bundles which are present here. They are also myelinated, and this time the myelination is given to them by the oligodendroglial cells. So, inside central nervous system it will be given by oligodendroglial cells, and these bundles which are running inside the central nervous system within the white matter, these are called tracts, and these are called the tracts because these tracts are going from below upward. You can call them ascending tracts. And similarly, from the higher centers the tracts can come even downward also, again myelinated, and myelination coming from oligodendroglial cells, coming from Neuro Epithelium. So, see, this is now the descending tracts, and these descending tracts, why they have come down here? Because they want to actually modulate, or they want to give wisdom to these cells. These are the motor neurons, like here you see there. So, they will actually be like here. So, these tracts are the descending tracts, and these will relay into these neurons. We say these are the descending tracts. Their cell bodies are lying in the higher centers, and they are now coming down and will give wisdom to these neurons which are lying here in the gray matter of the lower centers. So, that means this motor activity which is being carried out here for your effector organs will in fact be influenced by two different neurons. One neuron group that is actually lying in the higher centers, and second neuron group which is lying in the lower centers, like in the spinal cord and the brainstem. That is why this is called upper motor neuron, whose cell body is lying in the cerebral cortex, this time the motor cortex. And then its axon bundles are coming down within the white matter of various parts of your central nervous system until they will reach here to the motor areas of either brainstem above or spinal cord below, in their nuclei, in their specific nuclei. So, these are the tracts, and these specific nuclear cell bodies you will call as the lower motor neurons. So, that means upper motor neurons and lower motor neurons words are used for these group of neurons. And here you are actually watching this here. So, these are called the tracts, and we have told you that this is not just the neurites which are running in the central nervous system. There are neurites which are running outside central nervous system also. They are also myelinated. They are also ensheathed within the connective tissue. They are also the bundles. But such bundles which are running outside central nervous system, either myelinated or unmyelinated, you call them nerves. You call them nerves here, nerve fibers, nerves. But similar thing which is running inside the central nervous system, either going up or coming down here, again myelinated or maybe some unmyelinated also, you call them tracts. So, the tracts and nerves, they are the bundles of the fibers, standing outside or inside, located outside or inside. And there is the ganglion and the nucleus. If the ganglion, if the discrete collection of the nerve cell bodies in form of collections here, outside here, you see these are called the neurons here, these are called the ganglion. But if these type of discrete collections are lying inside the central nervous system in the gray matter, you call them the nucleus. So, nucleus and ganglion should be differentiated, tracts and nerves can be differentiated. Now, after these basic are the fundamentals of the nervous system, in the next classes we will be actually discussing here various parts. I have already told you in the central nervous system we have only 7 parts, 7 components, without there is no 8th component at all. Spinal cord, this will be our topic. In the next class we will actually discuss here the spinal cord, and next will be the brainstem, in which medulla oblongata, second, third is pons, fourth is midbrain, we will discuss them together, and then fifth will be cerebellum, and then sixth is diencephalon, and seventh will be the cerebrum. These 7 components we will discuss separately, and then will be their peripheral nervous system, we will discuss their nerves. As far the spinal nerve is concerned, we have just to discuss one typical spinal nerve, and as if you have studied all the nerves. But as far the cranial nerves are concerned, all the cranial nerves actually they behave in a different way. But they are not actually, each cranial nerve will behave different ways, but we can say the cranial nerves, we can divide them in 3 groups now. The purely sensory, the purely motor, and the mixed nerves. Here you see that any spinal nerve, any typical spinal nerve will be motor and sensory both. You see their bundles are sensory as well as motor. But in case of cranial nerves, we have sensory separate, purely sensory, purely motor, and the mixed nerves. This will be inshallah our next class, and it is a spinal cord. And my advice is that any gross anatomy topic should actually be taken in 4 stages. Any gross anatomy topic, it is not just neuroanatomy, in any topic. The first will be the fundamental facts, the basic facts about that, basic knowledge about that organ. Number 2, the clinical correlates here, what abnormalities, what diseases will hit that organ usually. Number 3, the practice of the questions, how the questions will appear in the exam. Now, how I will, but these questions will be the clinical scenario type questions, usually in our local exams and our usually in other types of exams, or you say in USMLE, in PLAB and others, AMC, usually you have got clinical scenario type of questions, and these questions should actually be put and have good practice about that. But this practice will be possible only, and you will be able to feel yourself comfortable only when you have got the basic knowledge about that structure and clinical correlation, what abnormalities can happen with this, and then you will actually solve these questions and see and give the answer with explanation. We will guide you in these things also. And the 4th thing will be the case presentation. Present the case related to that organ's abnormality, and then present the case whole total presentation along with its history, examination, its investigation, and all these things, positive findings, everything. You have to present the case, and at the end of the case 6 to 7 questions you have to explain using your basic knowledge here. This is the 4th stage that you have to see in any topic. And whenever you will actually take any topic, you should have bit of knowledge, you see brief knowledge about its embryology and histology. I say brief knowledge. You do not have to go into huge detail about that, but you see that the brief knowledge about histology and embryology. I have told you again here, the first order neuron, sensory neuron, first order sensory neuron, this its shape was actually like this here. See, this is cell body, one fiber, I say one axon splitting into the 2. Such things are called, such neurons morphologically they are called pseudo-unipolar neurons. These neurons were called as multipolar neurons, the motor neurons, usually in the central nervous system here. Sensory neuron are motor neurons, they are multipolar neurons. These are pseudo-unipolar. True unipolar neurons, true bipolar neurons are like this. Cell body here, on one pole it will be dendrites, another pole it will be axons. These are called bipolar neurons. And these are called pseudo-unipolar neurons. Unipolar neurons usually they don't exist in vertebrates. It's only on one pole. These are unipolar neurons. These are pseudo-unipolar neurons. First order neuron lying in the ganglion, derived from neural cells. The first order neuron which will actually be connected to the sensory receptors. These are called the first order neurons, and first order neurons should always be located outside the central nervous system in form of a ganglion mostly, and they are derived from the neural cells, and they will always be morphologically pseudo-unipolar, except the sensory modalities which are related to the special senses. Means it will be smell or vision or hearing or vestibular system. These 4 special senses will have a first order neuron being bipolar. But the test, it will be again pseudo-unipolar. Test is also the 5th sense, you see that is the 5th special sense is the test. So, it will have the first order neuron or the first sensory neuron having its cell body standing outside the central nervous system in form of a ganglion. It will be pseudo-unipolar again, just like I say just like general sensations. In general senses, primary sensory neurons again in the ganglion is a pseudo-unipolar again. So, pseudo-unipolar, we have got bipolar. So, some bit of histology is important in these things. The questions come on there. Usually questions are asked on these things also. So this is something fundamentals, you see the basic information about how we will actually proceed and what will be actually our process through which we have to go through. 4 stages, remember always 4 stages: basic information, then is the clinical correlations, then will be the scenario type BCQs, a basic you see that's called the best choice questions here, and then will be the clinical presentation, I say case presentation. So this will be actually our class. So class is let's sign off here and class is dismissed.